Back to the deep dive, the show where we cut through the noise, stack up the sources, and extract the most impactful nuggets of knowledge to get you well-informed fast.
Today, we're plunging headfirst into the incredibly intricate yet profoundly important world of RNA.
Now, you probably know RNA as DNA's indispensable partner, right? The genetic messenger. But what's truly fascinating, and maybe less known, is that RNA isn't just a simple strand. It's a master of intricate folding.
Exactly. And that precise 3D structure is absolutely crucial for its function in, well, everything from developing new medicines to engineering biological systems.
Right, its structure equals function.
Totally. And this brings us directly to one of the, let's say, holy grails in molecular design, the RNA inverse folding problem.
Okay, inverse folding. So not predicting how a sequence folds?
No, that's the forward problem, which is hard enough. This is the inverse. It's about designing an RNA sequence that will fold into a very specific desired structure you've chosen beforehand.
Ah, okay. Designing the sequence for the structure.
Precisely. And this is a foundational hurdle, a bottleneck even, for creating the next generation of RNA-based technologies.
And when we say hurdle, we mean a significant one, right? We're talking computationally challenging.
Oh, very. Even NP hard, technically.
Think about it. The number of possible RNA sequences for a given length is just astronomical.
It explodes.
So traditional methods, it turns out, really struggle with that sheer scale and complexity, trying to find the perfect sequence for a target structure.
Yeah, they get bogged down, often stuck in suboptimal solutions, or they simply take an unfeasible amount of time, computationally speaking.
So not very efficient.
Not for complex targets, no. But what if we told you there's a novel solution, a true breakthrough that leverages the power of, well, something different, attention-free language model?
Attention-free. Okay, interesting.
That's what RWKV-IF brings to the table, and it's looking like a real game changer.
So your mission for this deep dive, if you choose to accept it, is to unpack the cutting-edge innovations that make this RWKV-IF so remarkably effective.
We'll explore its unique architecture, its ingenious strategies for guiding sequence generation.
And how it ingeniously sidesteps one of the biggest bottlenecks in the field.
Yeah.
Data limitations.
Yeah.
So if you're curious about how AI is revolutionizing the very fabric of molecular design,
then this deep dive is definitely for you.
Let's unpack this.
Okay.
So before we dive into the how, let's really underscore the why. Why is solving this RNA inverse folding thing such a big deal? What are the real-world stakes here?
Oh, the practical significance is immense, huge. When we can reliably design RNA molecules with precise structures, we unlock just a whole array of applications.
Like what? Give us some examples.
Okay, consider advanced therapeutics. Precise RNA folding is essential for designing things like aptamers.
Aptamers, those are RNA molecules that bind to specific targets, right? Kind of like antibodies?
Exactly like antibodies, but they're RNA. High affinity, specific binding, but potentially easier to manufacture. Or think of siRNAs, small interfering RNAs.
Yes, gene silencing.
Key players in gene silencing, used in drug development to literally turn off disease-causing genes. You need the right structure for them to work properly.
So medicine is a huge frontier here, but it goes even broader than that, doesn't it? Like synthetic biology?
Absolutely. In synthetic biology, we're not just talking therapeutics. Engineered RNA molecules can be designed to function as, say, sophisticated biosensors.
Detecting specific things in a cell.
Yeah, specific substances in a cell or environment. They can even act as complex regulatory circuits, controlling gene expression with unprecedented precision.
Wow.
And then there's nucleic acid nanotechnology, where rational RNA design forms the bedrock for constructing programmable RNA-based scaffolds. Microscopic devices, even.
We're literally building molecular machines out of RNA.
We're getting there. The possibilities for engineering biology are truly staggering once you can control structure.
But historically, getting these precise designs was, well, painful.
Painstaking is a good word, often relying on empirical methods, basically trial and error in the wet lab settings.
I can imagine. Sounds slow.
Incredibly costly, time-consuming, and inefficient. And often you had really low success rates. It's like searching for that needle in a haystack, but you have to synthesize and test each piece of hay individually. Not fun.
Yeah, not practical for complex designs. So that's where computational approaches stepped in.
Right. Early search-based methods, things like RNA inverse, which is part of the popular VNRNA package, or NRNO, they try to navigate this vast sequence space using what are called heuristic optimization strategies.
Heuristics. So clever guesses, rules of thumb.
Kind of. Educated guesses, clever shortcuts, trying to find good enough solutions quickly, often based on simplified energy models that estimate how an RNA will fold. They were steps forward, definitely.
But they had shortcomings, significant ones.
Precisely. While better than the wet lab, they still faced high computational costs and, crucially, poor efficiency for long sequences.
Because the search space just gets too big.
It explodes exponentially, even with heuristics. And a major problem was their tendency to get trapped in suboptimal solutions.
Meaning?
Meaning they'd find a sequence that folded mostly correctly, maybe one or two base pairs off, but not perfectly, and that significantly limited the diversity and the optimal functionality of the designs you could get.
Okay. And then deep learning arrived on the scene. Models like CentRNA, Lerna. Did they fix things?
They certainly improved things, yeah, offered a more data-driven approach, but even these more sophisticated models still struggled with really complex RNA structures.
Particularly pseudoknots, I read. What are those again?
Pseudoknots. You can think of them like an extra knot tied within a loop of string. They introduce these non-canonical base pairings that break the simple nesting rules most algorithms assume.
Making them hard to predict or design for.
Notoriously difficult. And critically, these early deep learning models were also highly dependent on data quality.
Garbage in, garbage out.
Essentially, if the training data wasn't perfectly representative or diverse enough, the model's performance would suffer. It wouldn't generalize well.
Which brings us back to RWKV-IF and its foundational innovation, this attention-free architecture.
Exactly. This is where it gets really interesting from an AI perspective. The core conceptual leap here is treating RNA inverse folding not just as a molecular design problem, but as a conditional language modeling task.
Conditional language modeling.
Yeah.
Okay, that's a fascinating reframe. So instead of predicting the next word in a sentence,
the model is generating an RNA sequence A, U, G, C, conditioned on a specific input. And that input is the target RNA structure.
Usually in that dot bracket notation you mentioned. Parentheses for pairs, dots for unpaired.
Yep, that's the standard.
Yeah.
So the model's job is to essentially speak the RNA sequence that perfectly corresponds to that structural sentence.
That's a really neat way to think about it.
It is. And the real innovation lies in the specific model architecture they chose. RWKV stands for receptance, weight, key, value.
Specifically the Eagle RWKV-5 version, I believe.
That's the one. Now, unlike the dominant transformer models, you know, the ones powering things like GPT RWKV is fundamentally attention-free. Yeah, it uniquely combines the best advantages of both recurrent neural networks, or RNNs.
Which are good at sequences, remembering the past.
Exactly. Good memory. And the parallel processing power you get from transformers. It's a kind of hybrid, but without the attention mechanism itself.
Okay, but why is attention-free such a groundbreaking idea for something like RNA design? What are the actual benefits over a standard transformer?
Okay, for RNA design, it's revolutionary for a few key reasons. Transformers, while super powerful, have this quadratic time and memory complexity. It scales with the square of the sequence length.
Which becomes a huge problem for long RNA molecules.
A massive bottleneck. RWKV, on the other hand, boasts linear time and memory complexity. It scales directly, linearly, with sequence length.
Huge difference.
Game-changing difference. This means it can handle efficiently long-range dependencies, which is absolutely critical for understanding and designing those extended RNA structures, where interactions might span hundreds, even thousands of bases apart.
Right, which attention struggles with efficiently at long lengths.
Precisely. Plus, RWKV enables lightweight inference for large-scale generation tasks. It's just much more efficient computationally when you need to generate and test many, many design candidates. It's a leap in scalability.
Okay, linear complexity, super efficient for long sequences, I get that. But how does it do that without attention? Let's get a bit more academic, dig into the mechanics. It has these time mixing and channel mixing blocks.
Absolutely. Let's break that down. The time mixed block is, well, ingeniously designed to capture position-aware temporal patterns across the sequence. Think of it as the model's memory unit, handling the flow of information along the sequence.
But sequentially, not globally, like attention.
Exactly. It processes information sequentially, integrating both immediate context and historical information through a clever recurrent mechanism. It uses this novel decay formulation, basically an elegant math trick, to ensure that information from the distant past smoothly fades but doesn't just vanish abruptly.
So it maintains a stable long-term memory.
Precisely, which is crucial for those long-range interactions in RNA. And there's a receptance vector within this block that acts like a dynamic filter. It effectively selects which pieces of historical information are most relevant for the current position. Very efficient.
So it's like each position is smartly deciding how much to remember from its past, adapting its memory without the massive computational cost of global attention.
You've got it. It's a very efficient way to handle temporal dependencies. And complementing this is the channel mixing block.
Okay, what does that do?
This block is crucial for enabling nonlinear interactions between feature channels. So while time mixing handles the sequence flow, channel mixing allows different learned features about the RNA at a given position to interact in complex ways.
Mixing information across features, not across time.
Right. It uses techniques like linear interpolation and a clever sigmoid-gated modulation to control how information flows between these different feature representations. It allows for highly expressive, nonlinear transformations of the data at each step.
Okay, that makes sense. And what about this token shift mechanism? That sounds like another interesting innovation.
Token shift. It's remarkably lightweight, but surprisingly powerful for temporal mixing. It's equivalent to a simple 1D causal convolution with a kernel size of 2.
Okay, what does that mean practically?
It essentially allows for a learnable linear interpolation between consecutive tokens or positions in the sequence. Imagine, for each little piece of information about an RNA base, the model can learn to blend in a little bit of information from the immediately preceding base.
The very local mixing.
Very local, very efficient, but it enables sophisticated capabilities, things like per-channel temporal blending, meaning different learned features can have slightly different short-term memories. And fascinatingly, it helps facilitate the emergence of induction heads within single layers.
Induction heads. That's a concept from Transformers, right? About pattern copying.
Exactly. It describes how models learn to efficiently copy and generalize patterns they've seen. Seeing this powerful pattern recognition ability emerge in an attention-free model through such a simple mechanism like token shift. Well, that's a testament to RWKV's design elegance. A significant academic insight, really.
That's a really deep dive into the architecture.
Yeah.
Very cool. But let's shift gears slightly. Building the model is one thing, but using it effectively is another. It's not enough to just generate an RNA sequence.
Nope.
It needs to be the right sequence. Structurally accurate, diverse enough to explore possibilities, and this sounds critical, biophysically meaningful.
Absolutely. You need sequences that could actually exist and function in a cell.
So this leads us to RWKV-IF's decoding strategy, which you said was also innovative.
Yeah.
What are the pillars of control they implemented?
Yes, raw generation isn't enough. You need control. RWKV-IF implements a sophisticated 3-pillar decoding strategy. First, there's top K sampling.
Okay,
Instead of just greedily picking the single most probable base, A, U, G, or C at each step, it smartly restricts sampling to only the K most probable options, say, the top 5.
and then picks randomly from those 5.
Essentially, yes. The probabilities of just those K options are recalculated, renormalized, and then you sample from that smaller, higher probability set. This technique is brilliant because it balances stochastic sampling and biochemical constraints.
Meaning it allows some randomness, some diversity.
Exactly. Controlled diversity, while still strongly favoring sequences that are likely to fold correctly according to the model.
Okay, that makes sense. Pillar 2, temperature control. I've heard that term in things like ChatGPT. How does it work here?
Very similar principle. The temperature parameter scales the logits, those raw output scores from the neural network, before the probabilities are calculated. It's basically a knob for randomness.
This lower temperature means
Lower temperatures, less than 1, make the probability distribution sharper. The model becomes more confident, more deterministic, leading to more accurate sequences that closely follow what the model thinks is best.
and higher temperature.
Higher temperatures, greater than 1, flatten the distribution, making less likely options more probable. This introduces more randomness and increases sequence diversity, so you can tune it, prioritize accuracy, or explore more varied sequence options.
Neat. Okay, and the third pillar sounds really specific to biology. GC content biasing. Why is that so important?
It absolutely is. This is a crucial innovation for biological applicability. The stability and melting temperature of an RNA molecule are heavily influenced by its ratio of guanine-cytosine base pairs because they form 3 hydrogen bonds instead of 2 like AU pairs.
So you need the right GC balance for it to work in a cell.
Often, yes. Many functional RNAs require a specific GC ratio range for stability and proper function. So this method applies a dynamic bias, let's call it A, to the logits of guanine G and cytosine C bases during generation.
How does the bias work?
If the GC ratio in the sequence being built so far falls below a predefined target GC ratio, the model gets a little nudge positive bias to select G or C next. If the current ratio is already above the target, it gets a negative bias, discouraging G and C.
So it actively steers the composition.
It effectively steers the nucleotide composition of generated sequences, ensuring they are biophysically meaningful. They don't just fold correctly in the model, but they also have properties like stability that make sense biologically. A very clever addition.
That all sounds incredibly well thought out. But, even with the best architecture and decoding, you need data, good data. Lots of it. And you mentioned RNA inverse folding has this notorious data bottleneck.
Yeah, that's historically been a huge problem.
What were the main limitations of the existing data set people were using?
Existing data sets suffered from several critical issues. One was just lack of scale, not enough examples. Another was insufficient structure and sequence diversity.
Meaning they didn't cover a broad enough range of RNA shapes.
Exactly. Some datasets, like Eterna bench centimeter, had relatively few unique structures, or others like bpRNA1 mater had many repetitions. This can hinder a model's ability to generalize to truly novel structures it hasn't seen.
And you also mentioned difficulty with low-cost folding evaluation.
Right. Some data sets rely on experimentally determined structures, but predicting the structure computationally, like with RNA fold for sequences from these data sets, doesn't always match the experimental structure perfectly, which creates inconsistencies if you want to use computational folding as your ground truth during training or evaluation. It's messy.
So RWKV-IF tackled this data problem head-on with a really innovative approach, it sounds like, constructing a large-scale synthetic training data set.
Yes, this was, I think, one of the most critical parts of their success.
How did they actually build the synthetic data set?
They executed this brilliantly and simply. First, they just generated 2 million randomly generated RNA sequences, just random strings of A, U, G, C.
Of varying length.
Yeah, specifically between 80 and 120 nucleotides for this study. Then, and this is the crucial step for quality, consistency, and scalability, they used a well-established computational tool, RNA-fold, from the VNRNA package, to computationally derive the secondary structures for all 2 million of those random sequences.
So they generated random sequences, then predicted their structures computationally.
Exactly. So now you have 2 million sequence structure pairs where the structure is guaranteed to be the one predicted by RNA-fold. This entire process is computational, making it massively scalable and allowing for easy, consistent, and super low-cost evaluation. Every single sequence structure pair is internally consistent by definition.
That avoids the whole messiness of experimental structures not matching computational predictions.
Completely.
And the most impressive part, maybe, is this proof of generalization. I mean, it's one thing to train a model on synthetic data, but does it actually work on real-world RNA problems?
That's the million-dollar question, right? And the answer was, fascinatingly, yes. That's the crucial, almost surprising finding. Despite being fully synthetic, generated from random sequences, models trained on this dataset generalize remarkably well to real-world RNA structures.
How do they show that?
They compared the statistical properties, things like structural complexity metrics of their synthetic training set with real-world test sets, and found they were quite similar. Even more compellingly, their analysis showed the synthetic data actually contain, on average, more challenging structures, higher average max depth, higher max span.
So the model was trained on harder problems than it might see in a typical real-world test.
Kind of, yeah. Which is great for robustness. This provides really powerful evidence that these random sequence-based datasets are viable for training complex inverse folding models. It effectively breaks that data bottleneck.
That is a truly massive win for the field, given how hard good biological data is to come by. Okay, and quickly, how did they adapt the standard RWKV training process for this specific RNA task?
It was a smart adaptation. Standard RWKV is often for free-form text generation. Here you have paired input, structure, and output sequence. So they trained by sampling by rows, treating each structure sequence pair as a distinct training example.
Keeping the pairs intact.
Exactly. And they also use a loss mask. They masked out the input structure part when calculating the error, ensuring the model's learning focus was solely on predicting the correct sequence given the structure. That's the core generative task here.
Makes sense. Okay, so we've got this novel attention-free architecture, clever decoding for control, and this ingenious synthetic data solution. the big question. Did RWKV-IF actually work better than the older methods? What do the results, the numbers, say?
Oh, the experimental results are truly compelling, night and day almost. RWKV-IF demonstrated really significant superiority over those traditional search-based baselines, like RNA inverse fold and nTRNA.
You mentioned a huge gap earlier.
Yeah, there was a huge gap in performance across the board. It wasn't just a small improvement. It indicated a real qualitative leap.
Okay, lay it on me. What were the specific performance metrics?
All right, let's break it down. First, average edit distance. This measures how different the predicted structure of the sequence the model generates is from the target structure you wanted. Lower is better.
Right. How much error is there?
Exactly. RWKV-IF achieved an incredible 83.4% reduction in this metric compared to the baselines. It brought the average edit distance down from around 4.8 edits needed to fix the structure to a mere 0.57 edits.
Wow, from almost 5 errors down to about half an error on average. That's huge.
It's a dramatic reduction in error. And then there's full match accuracy. This is even stricter. What percentage of the generated sequences, when folded computationally, perfectly matched the target structure?
The ultimate goal, really.
Pretty much. Here, RWKV-IF showed an astonishing 89.2% improvement over the baseline average. It jumped from the baseline managing about 37% perfect matches, 0.37, to RWKV-IF achieving over 91% perfect matches.
Over 90% success rate in getting the exact structure right on the first try.
Yeah.
Essentially, yes, based on computational folding. This means it's much better on obtaining a perfectly matched sequence, and crucially, it requires fewer attempts. You don't need to generate and test nearly as many sequences to find a good one, which dramatically reduces the computational cost for large-scale design projects.
Those numbers are genuinely incredible. What does this fundamentally mean for the bigger picture, for the field of RNA design, computational biology?
I think this work really highlights the immense potential of lightweight generative models in advancing computational RNA design. It's not always about bigger being better.
Right, the attention-free aspect.
Exactly. It opens up new directions for scalable and flexible biomolecular sequence generation. The efficiency of RWKV, with its linear complexity and lightweight inference, makes it incredibly well-suited for the kind of iterative sampling and optimization under biological constraints, like that GC bias, that are so common in real-world RNA design workflows.
So it proves you don't always need the absolute largest, most complex transformer model to solve really tough biological problems.
That seems to be the message here, yeah. Smart architecture and clever training can go a very long way.
That's a powerful statement in the current AI climate. Looking ahead then, what are the future frontiers? Where does this kind of work go next? What's planned for RWKV-IF?
Well, the researchers mentioned plans to incorporate additional benchmark data sets to further, rigorously evaluate the model's generalization across an even wider array of real-world RNA types and structures. Keep testing its limits.
Makes sense. Push it harder.
But the truly challenging and maybe most exciting prospect they mention is extending the framework to support RNA structures with pseudoknots.
Ah, the tricky knots again.
Exactly. As we discussed, pseudoknots are notoriously difficult for most existing approaches, both forward and inverse folding. But they're absolutely critical for accurately capturing functional RNA conformations in many cases.
So cracking pseudoknot design would be a huge step.
Massive.
Right.
Imagine the possibilities if we could reliably design RNA molecules with these complex functional knots for novel therapeutics or nanodevices or synthetic biology circuits. That would be a whole new level of control.
What an incredible deep dive this has been. We started by highlighting this RNA inverse folding problem, this really tough, computationally challenging task of designing RNA sequences to achieve specific functional structures.
Yeah, a core challenge in the field.
And we've seen how RWKV-IF, with its groundbreaking attention-free language model architecture,
That linear scaling is key.
its ingenious decoding strategies like top K, temperature, and that crucial GC biasing to ensure biological relevance.
Getting the biology right, not just the structure.
And its really innovative use of synthetic data to overcome that data bottleneck. How all that has led to a monumental leap forward.
It really feels like a leap, not just a step.
It's not just a small improvement. We're talking about, an 83.4% reduction in edit distance and almost 90% improvement in full match accuracy over traditional methods. That's transformative.
It's a powerful demonstration, really, of framing these complex biological problems as conditional language modeling tasks. And the specific innovations within RWKV, the time mixing, channel mixing, that token shift mechanism, They are genuinely key to its efficiency and its ability to handle those long-range dependencies that are so vital in RNA.
And then pairing that architecture with the controlled decoding.
Yeah, that ensures the biological relevance of the generated sequences. It really feels like a masterclass in elegant AI design meeting biological necessity.
It really makes you think, doesn't it? If we can design functional RNA with this kind of precision, using these lightweight, efficient, attention-free AI models, what other complex biological design problems could be unlocked?
Proteins, metabolic pathways.
Exactly. Proteins, entirely new molecular machines. Could similar language model approaches tackle those too? It feels like we're just scratching the surface of what AI can do in the vast, intricate world of molecular biology.
That's a profound question, and definitely one that the intersection of AI and biology is going to be grappling with very excitingly for many years to come.
It's indeed. A lot to watch in this space.
Well, thank you for joining us on this deep dive today.
My pleasure. Fascinating stuff.
Absolutely. Until next time, keep digging into the fascinating world around you.